You know, there is this scenario in medicine that just it drives doctors absolutely insane.
Oh, I know exactly what you're talking about, the ICU mystery.
Right. You have two patients, they're in the ICU, same age, same background roughly,
Yeah.
and they both get hit with the exact same infection. Let's say a really aggressive bacterial sepsis.
And the standard of care is identical. Same antibiotics, same fluid, same heme.
Exactly. But then the outcomes are just completely different. Patient A fights it off. It's rough, but they walk out of there.
And patient B crashes, multi-organ failure, inflammation just goes haywire, and they don't make it.
And for the longest time, the explanation was just, well, bad luck or, genetics.
It's always been the black box of immunology, right? We assumed the difference was in the patient's own cells. Maybe their heart was a bit weaker or their immune system was just a little sluggish.
But we've got a study today that suggests we've been looking in the wrong place. Or maybe we've been ignoring a huge third party in the room. We are doing a deep dive into a paper from Nature Microbiology that argues survival in sepsis isn't just you versus the bug. It's actually a three-way war that involves your gut microbiome.
This is a really, really significant paper. The title is Enterobactin inhibits microbiota-dependent activation of AhR to promote bacterial sepsis in mice. And I know, that is a mouthful.
It is a total mouthful, but once you crack it open, it's basically a spy thriller happening inside your own body.
That's not a bad way to put it.
I mean, we're talking about intercepted communications, secret weapons, and a molecule we thought we knew that turns out to be a double agent.
That's a perfect frame for it, actually, because at its core, this whole study is about signaling. It turns out your gut bacteria are constantly talking to your immune system.
Telling it what to do.
Telling it how to handle an infection. And the pathogen, it's actively trying to jam the signal.
Okay, so before we get to the signal jamming, let's set the stage. The researchers started with a mystery in the lab that basically mirrored that ICU scenario.
Yeah.
We're working with mice.
Right. Correct. And this is a crucial detail. They used genetically identical mice.
No clones.
Essentially, yes.
Yeah.
Same genes, same food, same sterile environment. And they infected them all with Serratia marcescens, which is a really common, nasty, hospital-acquired pathogen.
Okay, so if genetics were the main driver here, all these mice should react pretty much the same way?
You would think so, but they absolutely didn't. Just like the human patients, some mice cleared the infection and recovered. Others, they deteriorated fast.
And the researchers found a tell, something that predicted the outcome.
They did. They were tracking what they call the sepsis score, how the mouse looks, how it moves. But the real predictor was body temperature.
A fever.
The opposite, actually. In severe sepsis in mice, you often see hypothermia. Their temperature drops. And they found that at just 8 hours post-infection, when the mice still looked mostly okay.
Just 8 hours.
Yeah. The ones that were going to die had this sudden, sharp drop in core body temperature. It was like a crystal ball.
Wow. That's eerie. So the die is cast at just 8 hours. The researchers see this split, survivors versus non-survivors. And now they have to figure out why.
Right. And since the mice are genetic clones, the next logical place to look is the microbiome.
The gut bacteria.
Exactly. And this is where most studies like this would, hit a wall. Usually you do something called 16S sequencing.
Which is like a roll call, right? An attendance sheet for bacteria?
That's a great way to put it. You're just listing who's there. You've got E. coli present, lactobacillus present.
Okay. So they do the roll call for the survivors and the non-survivors.
And the lists were basically identical.
What? So it wasn't that the survivors had better bacteria?
Nope. The attendance sheet was the same. It wasn't about who was there. It was about what they were doing.
Ah, okay.
So instead of just looking at DNA, they switched to metabolomics. They analyzed the actual chemical soup in the gut. All the metabolites these bacteria were pumping out.
It's the difference between knowing someone clocked in for work and knowing if they're actually, doing their job.
Precisely. And that is when the aha moment hit. The chemical profiles were completely different. The survivors were just awash in a specific class of molecules called indoles.
Indoles. Okay, what are those and where do they come from?
They come from tryptophan.
The Thanksgiving turkey amino acids.
Yeah.
The thing that supposedly makes you sleepy.
The very same one. That nap thing is a bit of a myth, but the biology here is very real. Tryptophan comes from your diet. Your body can't make it.
Right.
And certain good bacteria in your gut have an enzyme that takes that tryptophan and breaks it down into these indoles.
So the survivors had a busy factory floor of bacteria just churning out these indoles, and the non-survivors didn't.
The non-survivors were depleted,
Yeah.
very low tryptophan, almost no indoles in their cecum, which is the start of the large intestine,
Yeah.
and crucially also low levels in the peritoneum.
The belly cavity, where the infection was actually happening.
Right, where the battle was raging.
Okay, so we have a strong correlation. Survivors have indoles, non-survivors don't. But that's not causation. How do we know the indoles are actually what's saving the mouse?
And that's the whole second part of the paper, the mechanism. And it's fascinating. It turns out our immune cells, specifically macrophages, the big eaters that gobble up bacteria, they have a receptor inside them called AhR, the aryl hydrocarbon receptor.
I always think of receptors as like a lek or an antenna.
Antenna is the perfect word here. Think of AhR as an antenna sitting inside the macrophage. When an indole molecule, the signal, binds to this antenna, it tells the cell to do two things at once.
Okay, what are they?
First, it makes the macrophage more bactericidal. It just gets better at killing the invading Serratia. They proved this with a gentamicin protection assay.
Whoa, break that down. Gentamicin is an antibiotic.
Yes, but it's one that cannot get inside a human or mouse cell. So you put your macrophages and your bacteria in a dish, let the macrophages eat. Then you wash everything with gentamicin.
So any bacteria left floating outside get killed.
Exactly. The only ones that survive are the ones that have been successfully eaten and are now safe inside the macrophage.
So more survivors means the macrophages were hungrier.
Precisely. And when they added indoles, the macrophages ate way more bacteria. But here's the second thing it does, and this is absolutely critical for sepsis. It changes the type of immune response.
How so?
It shifts the macrophage from an M1 phenotype to an M2 phenotype.
Okay, I remember this from biology. M1 is the flamethrower mode, right? Just scorched earth, kill everything.
That's it. M1 is super inflammatory. It kills bugs, but it creates a ton of collateral damage to your own tissues. In sepsis, that M1 response, that cytokine storm, is often what actually kills you.
And M2 is what?
You're a repair crew.
The repair crew, exactly. M2 resolves inflammation, it promotes healing. So this indole signal is incredible. It tells the immune system, kill the invader, but for goodness sake, put away the flamethrower, don't burn down the house.
That's a level of nuance I didn't think the immune system had. It's disease tolerance. It's the definition of it. Tolerate the bug long enough to clear it without the inflammatory response killing you first.
And they proved this wasn't a coincidence, that AhR was the key.
They did. They used mice that were genetically engineered to not have that AhR antenna on their macrophages.
So they snipped the wire.
Snipped the wire. And even if they pumped those mice full of indoles, it didn't matter. They died.
Because a key, the indole, had no lock to fit into.
Yeah.
The message had nowhere to go.
Exactly. The signal must go through that specific receptor.
Okay, so up to this point, it sounds like a nutrition problem. Eat more turkey, make more indoles, survive sepsis. But you mentioned a villain. You said the bacteria were jamming the signal.
Yes. This is where the paper just turns into a full-on detective story.
Yeah.
The researchers figured, wait a minute, this pathogen isn't just a passive punching bag. It's fighting back.
How did they figure that out?
They took the liquid that the bacteria were growing in, they call it the supernatant, and they filtered out the actual bacteria. So all you have left is the broth with whatever chemicals the bugs secreted into it.
Okay, the chemical soup.
And they added that soup to the immune cells along with the good indoles that should have been turning on the protective signal. Nothing. The signal was blocked, the AhR receptor didn't activate, the macrophages were stuck.
So something in the water is jamming the antenna. How did they find the culprit?
Well, first they filtered the fluid by size,
Yeah.
and they found the jamming agent was a tiny molecule, less than 3 kilodaltons. Then they used mass spectrometry and these really complex molecular docking simulations.
Computer modeling.
Yeah, 3D modeling to figure out the structure. And the prime suspect was Enterobactin.
Enterobactin. Okay, I've heard that name before, but not like this. Isn't that a siderophore? Something bacteria use to steal iron from us.
Yes, that's its day job. We know that. Bacteria need iron to grow. Our bodies are really good at hiding it, so bacteria send out Enterobactin like a little grappling hook to snatch iron and bring it back.
But this study says it has a side hustle.
A very, very sinister side hustle. It turns out that Enterobactin shape is just similar enough to the indoles that it can also bind to that same AhR receptor.
Oh, I see where this is going. It fits in the lock.
It fits in the lock, but it doesn't turn the key. It's like putting gum in the lock.
It's competitive inhibition.
It's the perfect gum in the lock. It jams itself into the AhR receptor, and it just sits there. So when the good indoles arrive to deliver their life-saving message,
The door is blocked. They can't get in.
they can't get in. The calm down signal is never received.
That is just It's incredibly sophisticated. The pathogen is actively blinding the immune system to keep it in that self-destructive M1 flamethrower mode.
Which creates chaos, damages the host, and ultimately gives the bacteria a better environment to thrive in.
Is this a one-off trick just for this Serratia bug? Or is this the common playbook?
That was our next question, and it's a big one. So they tested other pathogens that they isolated from actual ICU patients. Things like Klebsiella oxytoca, Klebsiella pneumonia, even a fungus, Candida albicans.
But did they do it too?
They all did it. They all produced something that inhibited AhR. It looks like this is a widespread virulent strategy, a universal tug-of-war.
So let's just picture this war.
Yeah.
On one side, you've got team host. That's your gut bacteria, taking the tryptophan from your food and making indoles, the keys.
Right.
On the other side, team pathogen, pumping out Enterobactin, the gum for the lock.
And your survival literally depends on that ratio. Who has more molecules on the battlefield? If the indoles can overwhelm the Enterobactin, you live. If the Enterobactin blocks enough of those receptors, you die.
Which naturally leads to the next question. Can we rig the game? Can we help team host win?
They tried, and this part is really important for anyone thinking about, supplements. Their first idea was the most obvious one. Just feed the mice oral indoles.
Give them the keys directly. Did it work?
Nope. Failed completely.
What?
Your liver. First pass metabolism. You swallow a pill, it goes through your gut, and then it goes straight to the liver, which is our master detox organ. It saw the indoles and just chewed them up before they could get into the bloodstream at a high enough concentration.
So you can't just pop an indole pill and be fine?
Not easily, no. So they tried a different strategy. They went upstream. They supplemented the mice's drinking water with tryptophan, the raw material.
So how long?
For 2 weeks before they infected them.
So they were loading up the factory.
Exactly. They saturated the system with the raw material, so the gut bacteria had all the resources they needed. They started churning out massive amounts of indoles. And when those mice got infected,
They survived.
they survived at a much, much higher rate, even against a mix of different bugs. Because the sheer volume of keys, the indoles, simply outnumbered and outcompeted the gum.
It just flooded the system.
They flooded the system. And just to prove the point about location, they also tried one more thing. Injecting the indoles directly into the peritoneum, right onto the battlefield.
Bypassing the liver entirely.
And that worked brilliantly. It proves that if you can get the signal to the immune cell at the site of infection, you can stop the sepsis.
Okay, this brings us to the million-dollar question. This is all fascinating in mice, but are we mice? Is there any evidence this indole versus Enterobactin war is happening in humans?
There is. This is what makes the paper so strong. They didn't just stay in the lab. They analyzed stool and blood samples from human sepsis patients in the ICU.
And the data matched the mouse model.
It lined up perfectly. In the stool samples, the human patients who survived sepsis had significantly higher levels of indoles compared to the patients who died.
So the survivors had a healthier, more active microbiome that was producing the calm down signal.
That's the implication. And when they looked at the blood, they found that, in general, sepsis patients had depleted levels of specific indole metabolites compared to healthy people. It really suggests that losing this chemical shield is a major risk factor for a bad outcome.
This completely changes how I think about something like antibiotics. I mean, someone comes in with sepsis, first thing we do is hit them with broad-spectrum antibiotics.
And you have to. You absolutely have to kill the bug.
Of course. But what's the collateral damage? Those same antibiotics are nuking the gut microbiome. So we're killing the Serratia, which is good, but we're also killing the friendly bacteria that produce the very indoles we need to survive the inflammation.
You're destroying your own supply lines. You're taking the key factory offline at the exact moment you need it most.
It's a terrible irony. The treatment helps clear the bug, but it might be making you more vulnerable to the inflammation by removing that calm down signal.
It's a huge clinical dilemma.
So where does this leave us? I mean, are we going to start seeing tryptophan drips next to the saline in the ICU?
It's possible. Or maybe, specially designed indole-based drugs that can survive the liver. But I think the bigger takeaway is this whole concept of treating the host right alongside killing the bug.
Right. We're so focused on the invader. This paper shows that the host's ability to tolerate the fight is maybe just as important as the weapon you use.
And it brings up a really provocative final thought about what virulence even means.
How so?
We think of a bacterium as being deadly or mild on its own, based on its genes. But this mechanism suggests virulence is relative.
Relative to what?
Relative to your microbiome. If you have a gut that's thriving, full of these indole-producing bacteria, maybe Serratia isn't actually that deadly to you. You have the shield.
But if your microbiome is wiped out, maybe from a poor diet or from previous antibiotic use,
That exact same bacteria becomes a stone-cold killer because its signal jammer, the Enterobactin, faces no competition.
so we might be creating deadlier bugs not just by making them resistant to drugs, but by weakening our own internal allies.
Exactly, we're clearing the field for them, making it easier for their dirty tricks to work.
Wow. Well, it's a lot to think about. It kind of makes you want to go eat some fiber and thank your gut bacteria.
They're working hard down there, keeping the gum out of the locks.
Indeed. Well, that wraps up this deep dive into the microscopic war for the AhR receptor. It's a reminder that sometimes the most important conversation isn't the one you're having, but the one happening between your gut and your immune system.
Stay curious, everyone.
See you next time.